Good day, everyone. My name is Joy Concepcion from QPS. I will be your moderator today. On behalf of QPS's seminar series, I'd like to welcome you to the 5 S's of hybrid LCMS-MS methods: specificity, selectivity, sensitivity, strength, and speed, presented by Dr. Fabrizia Fusetti of QPS Netherlands. A few housekeeping comments before we begin. Everyone will be muted during the presentation. Please enter any questions under the control panel. If we cannot get to your question, we will respond to you directly after the seminar. This webinar will be available on demand to all registrants. We will send you an email once the recording is available. And now I would like to introduce our presenter, Dr. Fabrizia Fusetti, Director of Bioanalysis at QPS Netherlands. Fabrizia obtained her PhD in molecular biology and biotechnology at the University of Milan in Italy, after which she spent several years in academia and participated to numerous fundamental and applied research projects, mainly focused on the study of proteins. Her work resulted in over 50 peer-reviewed publications, and before joining QPS in 2014, she started and managed one of the proteomics platforms at the University of Groningen. She is particularly interested in the development of mass spectrometry methods for bioanalysis of biotherapeutics. And with that, I will turn it over to Dr. Fuscetti.
Thank you, Joy. Thank you very much for the kind introduction. Good morning, everyone. I'm very excited to welcome you to our first bioanalytical online meeting. And thank you all for joining us from all over Europe this morning. This new reality is nowadays encouraging us to find new ways to exchange information and to meet. And in the same way, the pace of drug development is constantly stimulating us to innovate and find new solutions, and to do that efficiently and fast. And this is what has inspired the title of this presentation. For the ones who are not familiar with this, the 5S methodology was originally implemented at Toyota in Japan to increase the efficiency on the workplace. And that was based on 5 basic principles. And all these basic principles start all with the letter S. So that's why the 5S. In an efficient and successful bioanalytical workflow, we also can recognize 5 basic principles. And if we use some creativity, we can also build in the 5S in this principle. So these are selectivity of the method, specificity, a high degree of sensitivity requirement, strength as a synonymous of versatility and robustness, and speed. As the faster methods are available, the more benefits this will bring to the drug development pipeline. Based on this, we would like to share with you our experience with these methods and give some examples on how a hybrid LCMS workflow can also satisfy these basic simple principles. First of all, I just want to go through quickly through the outline of this presentation. We will first talk about the key criteria that we consider for defining the bioanalytical strategy, and then go more into the depth technical details of a hybrid LCMS experimental setup, and what are the critical aspects for method optimization. And then after that, I will show you some examples of generic or specific methods, mainly focused on antibody-based biotherapeutics. And we will go through an example where we use the methods for candidate selection and preclinical Pka analysis, and another one where we brought that to a second step, and then finally to phase 2 clinical studies. But we will also touch on the multiplexing method for communication, and if there is time, also talk about how this method can be used in the early development of ADC, antibody drug conjugate, and how this can really make a difference in following enzyme replacement therapy development. So, oh, sorry. There's something wrong in this slide, I'm sorry, but all biotherapeutics are distinguished by a large degree of complexity. And by dealing with these methods, it's important to consider the degree of complexity and understand the biology. Most of these molecules have also very large molecular weights and present a large degree of complexity and heterogeneity. When going through the key criteria for defining the bioanalytical strategy, it's important to determine whether it is going to be more useful to use a ligand binding assay than a mass spectrometry approach. And in this case, we will look through the sensitivity and selectivity requirements. If there is expected or demonstrated cross-reactivity with endogenous molecules, and also what is the stage of drug development, and together with that, the regulatory aspect. Ligand binding assays have been considered until now the golden standard of bioanalysis for biotherapeutic. However, they often require the availability of specific antibodies and reagents and labeling for a perfect assay. And this has always a huge impact on timelines. And whereas sometimes a mass spectrometry method can be developed in a much faster way, without losing without losing any molecular information. In the next slide, you can I use this to show the variety and the complexity of IgG-based therapeutic. So starting from let's say a basic, simple IgG1, one can achieve a large degree of specificity by building different features on a common score, on a common scaffold. And in this way, it's possible to generate chimeras with multiple specificity. And as in this case, you can see all these multi-specific molecules. But with hybrid LCMS, one could even think of developing one method and being able with that one single method to describe the pharmacokinetics of all these molecules. This is in theory possible. And therefore, once we plan this method, we have to do that carefully and keep in mind in which stage this is going to be applied, if it is a preclinical or a clinical method, and if it's going to be, if selectivity is needed, or it can be also solved by a generic assay. And as for many different multi-specific molecules, these methods can also be applied for the Pka analysis of antibody drug conjugate, as they are also often based on an antibody. So now in the next slide, I will go through the generic experimental setup. LCMS methods can be of two types. You can have methods that target intact molecule, and there are bottom-up and other bottom-up methods where the proteolytic peptide is quantified, assuming that concentration of the proteolytic peptide is equimolar to the protein concentration in the sample. These methods go through proteolysis step, and I will go through these steps one by one in the next slide. Now, just a quick let's say, overview of the important crucial step of the workflow. The important part of a hybrid LCMS method is an affinity purification step that is often achieved for practicality with a ligand bound on magnetic beads. And this can be ready to use or customized depending on the type of assay that has to be developed. For antibody-based therapeutic, we can take advantage of the existence of molecules like protein A, protein L, or G, which bind to different parts of the antibodies in a very specific way. But we can also use much more sensitive purification methods by immunocapture, applying anti-Fc antibody or even anti-idiotypic antibody if they are available, or anti-payload antibody when dealing with antibody-drug conjugate. This is the place where functionality can be built into the method. So you can even think of doing functional pull-outs, keeping in mind what is actually needed to be measured at the end of the road, and then use a specific receptor, a ligand, or a cofactor in this respect. Afterwards, the sample goes through a chemical treatment, and then finally the protein will be proteolized. There are different proteases available, and depending on the scope of the project, it will be necessary to choose the protease very carefully. Most of the time we can use trypsin, but there are many other equally good and robust, maybe more expensive proteases available if trypsin is not suitable for achieving the desired selectivity. And after that, the peptide will go into the mass spectrometer, and then eventually at that step we will select a quantified surrogate peptide that will be the one that will determine eventually the concentration of the molecule in the sample. But we can also take other qualified peptides along that will become very useful during troubleshooting or to draw some metabolism conclusions. What really made a difference in the last years in the advance of this type of method is the possibility to use stable label internal standard to monitor the whole workflow. The use of an internal standard is a key factor in all mass spectrometry methods, as that is allowing to monitor for the variability in the extraction steps. If an identical protein is available as in the labeled, stable label version, and nowadays many antibodies are being synthesized with stable label isotopes present in the molecule, it is, let's say, that is the best solution, because that can be introduced in at the beginning of the workflow, and we'll be able to monitor all the steps of the workflow. But if that is not available, an alternative, sometimes equally good solution, is the use of a stable label extended synthetic peptides that will generate an equal surrogate peptide upon cleavage. So it will be not monitor this affinity purification, but it will be able to give us information on the yield of the and variability of the last part of the method. As well, it is possible to introduce one surrogate peptide, identical surrogate peptide, and the label version at the last step. Well, as I said here, I just wanted to highlight how these two factors, so which is used, which ligand is used, and which quantifier surrogate peptide is used for determining the concentration at the end, will also might also give functional and additional molecular details to the bioanalytical determination. The sample in mass spectrometry, less is more. We tend to use a smaller amount of a smaller volume of sample, most of the time lower than 10 microliters. Less sample will give less noise, less background, less interference, and then more of a target response. And you can even imagine that if you can use less than 10 microliters or even 10 microliters of sample, you could do a single mouse experiment with this workflow. As I said before, what is crucial in the affinity purification is to choose the right pull-out strategy, because that is going to define the method specificity and eventually the result at the end of the road. And this is this is the first selective step of the method. In addition, and highly sensitivity and selectivity of this method will also result in a much higher sensitivity, because also in this case it will reduce interferences. The more selective the affinity purification, the less interference from the background, and higher the enrichment. The sample treatment should be tailored to the peptides that will be monitored. It's a pretty standard step, but it can be very critical, especially because one should keep in mind the molecular stress that can be introduced at this point and later on during the proteolysis. It can also be very critical for the analysis of ADCs, where it can have also possible effects on the linker conjugation chemistry, and then it should be carefully taken into account for stability assessments. In the proteolysis, probably the most critical aspect is determining the sample protease ratio, because that will definitely influence the yield. And as for each step, the aim is to maximize recovery, the sample protease ratio, as well as all the environmental parameters that can influence the efficiency of the proteolytic cleavage should be taken into account. We also use the solvents and sometimes mass-compatible detergent to reduce possible non-specific binding of the proteolytic peptides. And then at the Lcms step, well, chronologically, as I'm going through the workflow now, this is the last step. Actually, this is the first step that is set up during method development. And optimization of this part will aim at getting the highest quality and reproducibility of the data in combination with an acceptable throughput. And then like all the rest of the steps, the key issue here is to reduce the background, resolve the analytes, and reduce the interferences. At this point, you can also include an additional step, an SPE purification that will then work as a second dimension for the liquid chromatography, and will also help and might help in increasing selectivity by further reducing the interferences from the background. And for the rest, this is going to be just an optimization of mass spectrometry, like for many other methods. So yeah, I want to show you how a mass spectrometry looks like, because this is the hardware of a typical quadrupole mass spectrometer. These type of instruments are built to carefully select from a complex mixture, and that is why they have been used as a workhorse for bioanalysis since many years. What actually happens here is that each second, every peptide that is eluted from a chromatograph, from the chromatographic column, will enter the mass spectrometer. And then at this point, there will be a selection, it's an ionization first, and then a first selection step where one peptide of all the many that enter the mass spectrometer will be selected. This will go through a fragmentation, and eventually what will be measured on the detector are all the fragments that are generated in the mass spectrometer. So the pair of this mass and the mass of the fragment will determine at the end the data that is going to be measured. So I think it's important to keep in mind what is measured in the experiment, and also think that although the optimization of this stage is very important, and working with a state-of-the-art instrument is definitely going to influence the quality of the data, you should also consider that everything that, so this instrument, these methods will always give you a result. So the quality of the data that is obtained at the end will strongly depend on the on how carefully the sample preparation has been optimized and what is actually putting into the mass spectrometer. Even before starting with the experimental part, there is a in silico analysis that needs to be done to determine the choice of the surrogate peptide that will be monitored in the method. What we normally choose is a peptide that has a length between 6 and 25th amino acid. Too short, it will not give sufficient selectivity, and it will probably also give cross reactivity from the background and endogenous sprouting. A longer peptide might be a challenge for the mass spectrometry determination. Other aspects that can be identified easily in the sequence, and although have to be at the end experimentally verified, can be hydrophobicity and let's say all the factors that could influence the ionization and the fragmentation abilities, and then eventually the sensitivity of the method should also be taken into account. As I said, yeah, cross-reactivity is very important, and that can also be tested with online tools like, for instance, BLAST is very useful to check if there is any protein that contains the same identical peptide. This is also the step in which a multiplexing method can be planned by selecting different peptides in the protein. The in silico proteolysis can also be done online, and then you can choose between different proteases, but also predict post-translational modification that could also affect the method by producing mass changes in the peptides that has to be measured. So also this has to be taken carefully into account. When developing antibody-based therapeutic, the normal, let's say, approach is to reduce immunogenicity by building, so by doing CDR grafting. This will result in a molecule that will be largely identical to a human IgG, but it will still carry the antigen binding site and the specificity of the original developed of the original antibody that can be developed in mouse or in different way nowadays. The diversity within these molecules and the common feature will be reflected by a difference in the sequence, in the amino acid sequence. And therefore, proteolysis of these molecules will generate different peptides, like in this region that corresponds to the CDR of these 3 example light chain sequence from monoclonal antibodies. But it will also generate common peptides. So depending on the choice of the peptides that will be used for the method, we can generate specific methods that will be then only able to recognize one of the 3 species, and generic methods that will be applicable for identification of many molecules of this type with different specificity. And this is also what will bring me to the first case study. And by going through some of our from some of the methods that we developed in the past years for PK evaluation, I want to give you some examples to show you how these methods can really be of advantage and benefit from preclinical to clinical development. This is one of the most interesting methods we have been developing. The bioanalytical challenges was to develop one method for candidate selection that was able to target 7 different versions of the same antibody molecule. And these were derived by either IgG1 or IgG4. They had a different structure, but all of them had the same specificity. Ligand-binding assay was not possible because specific reagents were not available at that time. And also an initial ELISA approach showed a very high cross-reactivity and background when applied to even to the preclinical matrices. Together with that, the sensitivity requirements were quite high as we were working with small animal, and also we had samples from different species, from different matrices, different species and tissues. So the sensitivity was really initial, but also the sample volume was not very high. And on top of this, the timelines were very tight. So for this method, basically all the ingredients for having for the requirement for speed and specificity and selectivity were present. So what looking at the molecule, we choose to do a purification with protein L, which is able to bind to the kappa light chain of the immunoglobulin, and so it will target this part of the molecule. And then in this way, we had just one affinity purification method for the different types. We choose for a Lc protease instead of trypsin, and selected for a 23 amino acid peptide from the CDR region. The species were red rabbit and non-human primates. As I said, we had tissue and serum. And at the end, we were lucky because the surrogate peptides that we choose in the CDR region was also the peptide that had the highest sensitivity. And because of its selectivity, the method could also be readily applied to other preclinical and possibly to clinical matrices. The statistics were, as you can see here, in the accuracy and precision were very tight. Just to remind you that for chromatographic methods, the requirements of the guidelines for accuracy and precision are that they should stay under the 15% for a small molecule, 20% at the LLOQ, and large molecules. For large molecule methods, 5% additional is allowed. And this is also bringing up often the discussion which criteria should be applied for this method. In this table, you can see that although the methods were only qualified, and so these numbers are not based on a large number of data points. The accuracy and precision are most of the time below 10%, and only at the LLOQ and 1 year, only 1 data point, is much higher than 10%. But still very reasonable precision and acceptable precision and accuracy. So this gives you the idea of the quality of the data. And just to show you how the result of this project, I would like to share this Pka Graph, where that basically contains some of the Pka evaluations for one of the matrices, including 5 of the ligands that were targeted. And then you can see that the methods are very are able to describe reliably the Pka behavior of this molecule. In blue are the smallest version, so the Fab fragment. In red, you can see the intermediate version, and in this one where the different Pka come together is for the full-length immunoglobulin that show that the concentration is maintained higher for a much longer period of time. In yellow here, we overlap the results from the ELISA, and although this method was also able to describe the Pka, let's say the Pka behavior of the full-length molecule, it shows a much higher spread than the mass spectrometry data, which is much tighter. And that is basically the result of the statistics that we saw in the table in the slides, one slide before. This is another example and could be theoretically the next step of the method that I showed you before. Also in this case, we were dealing with a Fab fragment that presented a high endogenous cross-reactivity. Because it was not a full-length immunoglobulin, we choose to purify this with protein L because of the binding to the kappa light chain. And the sensitivity requirements were not extremely high, around 3 nanomolar, compared to the method before, where we were able to reach a 5 times lower concentration than this. And also in this case, we selected for a specific method targeting 25 amino acid peptides from the complementary determining region of the Fab fragment. As the internal standard, we use trastuzumab as for this, as it was useful to monitor the whole sample preparation behaving in the same way as the molecule, but as we would not be able to find equivalent labeled peptides in any of the commercially available antibodies, we decided that we tried to see if monitoring an analog would still be sufficient to correct for the variation in the method. We successfully developed a method that was that also validated in 3 different matrices. First in non-human primate plasma, and then in human plasma. As the sponsor was also concerned about renal clearance, the method was also validated in human urine. And the transfer went really seamless and smoothly, showing exactly the same quality as the other methods. I here show you only the statistic of the for the human plasma validation in human plasma, but you can believe me, and we can show you in another moment that this quality was also present, maintained in all the other methods developed. Here you can see that all the statistics are below 10%, which is also an indication of the reliability of the method and the quality of the accuracy and the quality of the data that is produced. As also for the other, I didn't really spend time on talking about that, but these methods show also extreme higher linearity over a large dynamic range of about 3 orders. At the end, we have been developing this method for preclinical tox studies, and then this method is being now used for phase 2, and has accompanied the development of this molecule from the early stage to phase 1, and then now we're still measuring samples coming in from patients tested from 2019. And every time patient samples come in, we can just pick up the assay and apply it, knowing that the result will be identical to the previous time, without having to be concerned about reagents and validation of critical reagents. One other important application for this method is for enzyme replacement therapy. Often, recombinant proteins that are used for enzyme replacement carry only small differences compared to the endogenous molecules. In this case, I cannot share any information on the protein, but this is the real structure of the protein. The difference between the recombinant and the endogenous is just in 2 point amino acid mutations. And we have used this method for preclinical PK evaluation, and we will transfer it also for clinical bioanalysis. Because of the large degree of identity between the recombinant and the endogenous protein, it is not possible to distinguish these two with a ligand binding assay. There will be no reagent able to distinguish between the 2 point mutation. And on top of that, the sensitivity requirements are quite high. So we were asked to go below 10 nanogram per milliliter, and so below 150 picomolar. And to be able to monitor the endogenous and the recombinant protein at the same time, it was also important to have a method that could multiplex. So to have a simultaneous analysis, simultaneous quantitation of both forms. In this type of method, you also need to be lucky that nature is coming towards you. And we were, so the let's say, introduction of this 2 point mutation changed completely the proteolytic pattern of the molecule when it was after trypsin cleavage. So here you see a tryptophan was mutated into an arginine, and making it this peptide unique for the endogenous protein. And in this case, an arginine was mutated into a serine, producing again a unique peptide for the recombinant. And a selection of the best, of the highest sensitive method for this and this peptide has eventually produced the method that we are using now. For here, you can see that there is no cross-reactivity when we use apply the method for the recombinant and the endogenous protein. The blanks are clean. And as soon as we spike in the molecule, even at 5 nanogram per milliliter, so half of the target LLOQ, we can see a clear peak. Although these two seem to be at the same place, if you look carefully into the retention time, you can see that one has a retention at 7 minutes and one has a retention of 4 minutes, which means that these two are clearly separated into the chromatography, and there will be no crosstalk between the recombinant and the endogenous protein, although they are purified with the same immunocapture based on commercially available antibody. The linearity of the method is great, and although this is based on very few data points, you can see that also in this case we can count of a high accuracy and precision in this method. And like this, a method can be set up for other recombinant proteins used for in gene therapy development or in enzyme replacement therapies. Now I would like to show an example on which we use a multiplexing assay for a clinical pharmacokinetic analysis, where trastuzumab and pertuzumab were co-administered in a late phase study in a patient with I'm sorry, but my screen is frozen. It went well in all the rehearsal, and now I knew that something could happen. But so the occurrence of co-administered therapeutics is becoming more and more used in the clinic to increase the survival of oncology, the survival for oncology patients. Trastuzumab and pertuzumab are often used in combination for the treatment of metastatic tumors, but they both bind to the same receptor, the same HER2 receptor, and they have a complementary mechanism of action. One will bind to the dimerization domain, and the other one will block the functionality of the receptor. So as they are both binding to the same ligand, they are also not easily to distinguish by ligand binding assay, and especially not when we want to have a simultaneous quantification in the same sample. In this case, as often happens for a patient study, the sample is very precious, and the volume of the sample available is always very low. So we were again facing the same challenges as we had for the preclinical methods. In this slide, you can see again the linearity of the response of the two peptides chosen for trastuzumab and pertuzumab. Also in this case, the peptide had to be chosen from the CDR region that was specific for both molecules that are 98% homologous in sequence, amino acid sequence. So there is not much, there are not many regions in the molecule to choose for a peptide. So we had to do with what was available. And you can see here that again, in the blanks, there was no cross-reactivity, and the two peptides that were chosen have different retention time and can clearly be distinguished. And the statistics for the method are also very good and acceptable within the criteria suggested by the guidelines. The ranges went from 250 nanogram per milliliter to 25,000. So here again, 3 orders of magnitude, and the LLOQ was chosen for both molecules based on what it was already, on the data that was already available from previous evaluations of the singular therapeutic from ELISA assays. So looking at this, we could have gone maybe a little bit lower, but we didn't choose to do that. Well, here you can see that for each patient that participated to the trial, the method was used to describe the PK with 2 samples. This work has been published last year, and the method has been used for a study that lasted for more than 2 years and enrolled about 40 patients. So it is really actually used in the clinic. And if we look at the mean true and peak concentration as they were measured in serum, we can see that the C maximum did never exceed 170% in or 200 microgram per milliliter in for trastuzumab, and it was below 400 for the pertuzumab. At the same time, the C min was never lower than 20 micrograms per milliliter, showing that the range that was chosen for the validation of this method is fit for the purpose of the study. And it also means that it's not always necessary to push sensitivity if the method is sufficiently sound to describe or to fit the purpose of the study. And in this case, all data were fitting in the middle of the validated assay range. Well, I will now go very quickly through the last example. Antibody-drug conjugates are traditionally bioanalysis of antibody-drug conjugate is traditionally done by ligand binding assay in combination with mass spectrometry for the determination of the concentration of the conjugated antibody, total antibody, and for the determination of the payload, free payload, and also complemented with antibody-drug anti-drug antibody assays for immunogenicity assessment. But as demonstrated for the previous example, you can also think of developing a method that is suitable for the quantification of the total antibody. In this case, we develop a generic method choosing a peptide from the heavy chain and an extraction with protein A magnetic beads, which was sufficiently selective for a preclinical matrix. And we obtain a method that is able to quantify this antibody-drug conjugate with an LLOQ of 200 picomolar. As the surrogate peptide is chosen from the heavy chain, from the constant region of the antibody, you can also imagine that this method could be applicable for many other molecules with different payload of different specificity, which still maintain this surrogate peptide conserved. And to add another level of versatility to this type of method, you can even imagine that with the same sample, you can extract before extraction and determine the free payload by LCMS, by traditional small molecule LCMS method. And then depending on the nature of the conjugation linker, you can obtain also the total payload and measure that with LCMS. And so by doing this, this workflow could be comparable and produce comparable results to the 3 assays that I showed you in the slide before. So this brings me to the end of the presentation. I hope it was informative for you on how hybrid LCMS can be used in modern bioanalysis to build in selectivity and specificity, but also sensitivity, and give robust and versatile assays, and also allow us to meet the requirement for speed and fast timelines that are dictated by the modern drug development pace these days. And with this as the last comment, I would like to thank you all for your attention and for following us today. Thank you.
Thank you, Fabrizia. We will now take some questions from the audience. So the first question is, can the same hybrid LCMS-MS method be used to quantitate different IgGs, or different methods for IgG1, IgG2, and IgG4?
Well, if I understand well, the question is whether you can target more isotopes of more antibody isotopes with one method developed, with one hybrid method. And well, I think in theory, if you really know, if you can point out common features in all these isotopes, and these are maintained for all therapeutics, it's possible in theory to develop one method that is able to that will be able to quantify all of them. You might have to compromise in sensitivity requirements and cross-reactivity, and then at certain points you might want to move from this possible generic assay to something that is more specific. I think it's realistic for IgG1 and IgG4, which are the most used nowadays for antibody drug, for antibody-based therapeutics. And also, you know, the sequence of these molecules are known, but yeah, I don't think it will be possible to have one universal method for all isotopes. I think we can.
Will monitoring of many peptides decrease the quality of the data?
No, not with the modern mass spectrometers. Nowadays, the detectors are so sophisticated and fast to be able to collect many data points in a very short period of time without losing quality in the data. And so that's why it's actually advisable to include more transition, so more peptides in the method, because for compliance, it will be important. So the quantification should be based on one data point per sample. So for quantitative purposes, you will only use the surrogate peptide, one peptide per molecule, more peptides if there is a multiplexing assay. But it is advisable to also take along what we call the qualifier peptides. So there are other proteolytic peptides that for different reasons are maybe not suitable for the quantification, but they are very informative for troubleshooting during method development, and can also be used to obtain metabolic information. And as the collection of more data doesn't cost any time or any additional effort, I think it's good to take as much as possible, and being able afterwards to go back and look at data if other questions arise. after the method is developed, rather than having to reanalyze everything.
Got it. Okay. Will immunogenicity effect be visible if PK is determined by LCMS?
Yeah, this is a very good question. And I think it has to do with the fact that ligand binding assays, so with ligand binding assays that are used for immunogenicity, one can clearly distinguish between can you repeat the question, Joy? I think I'm not sure I understood that correctly. Can you just say it once more?
Will you be able to see immunogenicity if PK is determined by LCMS?
Oh, yeah, no, okay, I understood. So yes, most of the time when you do a PK analysis, you can, with ligand binding assay, you can see if antibodies, anti-drug antibodies are present that interfere with the assay, with the bridging assay. And I think if you, and you can see a difference if you do acid dissociation, so if you eliminate this interaction compared to when you do not do acid dissociation and you maintain the interaction between the drug and the anti-drug antibodies. In theory, you could do the same also with mass spectrometry if you are with these hybrid methods. If you are able to distinguish between the two forms, also in the affinity purification step. So if your reagents are able to see the difference between to select for the complex, for the bound drug compared to the free drug, and by repeating the experiment before and after acid dissociation, I think you might be able, even with these methods, to get some information on that, on the presence of anti-drug antibodies. And you might even be able to get some more molecular details compared to a ligand binding assay. Although these are, I don't think is also, you know, accepted from a regulatory point of view, but I think it's worthwhile to investigate if there is a suspicion that the immunogenicity can arise.
Okay. How low can your LOD or LOQ be? Can you get into the picogram range with LCMS?
Yeah. Well, it depends how big is your molecule. So I think the maximum we had demonstrated with these examples today was in the order of the 80 picomolar, so the 5 milligram per milliliter for a 70 kilodalton protein, 5 nanograms per milliliter for a 70 kilodalton protein, depending also on the length of the peptide, is equivalent to about 80 picomolar. So the sensitivity depends on how you look at it. If you look at it from the protein point of view, then it depends on how big is the protein. Because at the end, what you quantify is a peptide. The quantification of this peptide does go down to the picogram per milliliter. But when you translate it to the molecule, well, yeah, it will depend on that. And it will strongly depend on the physical chemical feature of the peptide and how well it ionizes in the mass spectrometer, and how much retention you have on the column, how sensitive the affinity purification is going to be. So I think theoretically it can be assessed, let's say it can be predicted, but it eventually should be assessed experimentally with a feasibility assay. And what we normally do, we just develop, look at the maximum limit of detection of that peptide in the mass spectrometer. if necessary, by also using a synthetic peptide. And then it's question of aiming to the maximum recovery and maximum reduction of the background interference.
Okay, one last question. Did you ever try to quantify antibodies by analysis of light chains using high-res MS in order to overcome the drawbacks of surrogate peptide? For example, when it's difficult to find a specific and sensitive surrogate peptide.
Well, we haven't tried, we haven't done it here, and in a regulated environment. But yeah, I think it's definitely possible as an alternative, although the sensitivity is often much lower, at least nowadays, with a technology available. It's much lower than for a surrogate peptide, so less sensitive assay. And well, you get an amazingly high amount of data. So you are still, you need to deconvolute the data. And although it is certainly very informative, it is still far from being accepted as a routine regulated approach.
Okay. Well, thank you, Fabrizia, for your time today. We really appreciate it. QPS would like to thank everyone for their time and attention today. You should receive an email with a link to the recording of this webinar. Meanwhile, stay safe and take good care. Have a great day, everyone. Bye-bye.
Bye-bye. Thank you.